Just wait, I will tell. Yeah, Vibila ma'am, we can proceed.
Everybody, I'm happy to introduce our next speaker.
Yeah. Yeah. Ma'am, is it audible?
Yes.
Yeah, Ms. Jagrati is a Mary Curie Research Fellow at Plymouth University in UK. She completed her MTech Nanotechnology from Amity University, Delhi, India. And today she'll be talking about dual layer graphene and reduced graphene oxide based electrochemical biosensor for label-free detection of beta amyloid biomarkers. So I think Jagrati, you can start your presentation.
Okay. Hello, everyone, whoever is listening. Today, I'm going to be giving a presentation on dual-layer graphene and reduced graphene oxide biosensor for the label-free detection of beta amyloid. Beta amyloid is one of the well-known biomarkers of Alzheimer's disease.
Jagrati, please share your screen first, then you can start.
I'm sorry?
Yeah, please share your screen.
My screen is not shared?
Yeah.
No.
Okay, I apologize. I hope everybody can see my screen.
Yeah.
Okay, so as I was saying, I'm going to be giving a presentation on a dual-layer biosensor that we made for the detection of beta amyloid, a well-known biomarker of Alzheimer's disease. This project is a part of a blood biomarker-based diagnostic tools for early-stage Alzheimer's disease, a European Union project that is currently functioning until the end of 2020. If you want, you can go to our official website and get more information on it. So yeah, let's just start. Okay, so just an overview of today's presentation. I'll give you a brief introduction on what is Alzheimer's disease and what happens inside the brain of an Alzheimer's patient. Then we'll talk about the conventional detection methods. Then what is a biosensor? Then just basic information on electrochemical biosensors that we are talking about in today's presentation. And then we'll be coming on to the dual layer biosensor that we prepared for the detection of beta amyloid and what are the advantages and the conclusion. So by definition, term dementia is used to describe a group of symptoms associated with decline in memory or other thinking skills, which are severe enough to reduce a person's ability to perform everyday activities. So basically, it's an umbrella term to describe a range of symptoms associated with cognitive impairment. And although there are different types of dementia, as you can see here on the screen, but Alzheimer's disease is the most prominent one and accounts for almost 50 to 75% of the total dementia cases. And if we look at the stats, Alzheimer's disease worldwide projection shows that currently 40 million people in the world are affected with dementia, Alzheimer's disease particularly, and it's going to rise up to 106 million by 2050. And despite this, it is the only major disease which does not have an effective strategy to cure or even slow down the disease progression. So what goes inside the brain of a Alzheimer's patient? So what you can see here is a brain of a healthy patient, and then there is an Alzheimer's brain. So what happens in an Alzheimer's brain, there is a shrinkage of the brain that happens especially in the cortex and the hippocampal area. And this hippocampal area is usually involved in the key role of forming new memories, and the cortex region is usually involved in the thinking, planning, and remembering. And then there is, the ventricles, that is the fluid-filled spaces within the brain, they're also affected and they grow larger. And this usually happens because of the formation of plaques inside the brain of the patients, senile plaques. And these senile plaques are usually formed due to the accumulation and aggregation of a biomarker called beta amyloid. So largely at the moment, the Alzheimer's disease diagnostics is based on the neurophysiological test, which are again based on clinical symptoms. And these clinical symptoms occur when the brain of the patient is already damaged and the irreversible neuronal loss has started. And although there are alternate techniques, such as the one you see here based on MRI and PET or CSF sampling techniques, but these are really invasive. They can be invasive, like CSF sampling procedures called the lumbar puncture is invasive protocol. And these MRI, PET scanners are really expensive. And Alzheimer's disease being a global issue, a lot of clinics receiving the patients may not have access to such sophisticated instrumentation. And therefore, a point of care technology based on point of care technology can really help and can revolutionize the way the diagnostics is currently performed. And in this project, we're only trying to work on the blood biomarkers because blood sampling is far less invasive compared to CSF sampling techniques, and it can be employed to larger communities easily. So what is a biosensor? Basically, biosensor is an analytical tool which converts the interaction between a biological sample, which converts the interaction of a biological sample with a sensing surface into an output signal, which can be read by the users. So we have a biological sample, which in our case is usually blood sample or plasma sample particularly. And then there is an analyte of interest that you can see here. At this, then we have a biorecognition element on our sensor, which is very specific to this analyte of interest, and it only recognizes that. And then there is this transducer element, which will convert any interaction between the biorecognition element and analyte into an output signal, which can then be read by us and be evaluated. So in this presentation, we're basically only focused on electrochemical biosensors. Electrochemical biosensors convert the interaction between the analyte and the sensor surface into an electrochemical signal, which is based on generation or consumption of electrons. And these flow of electrons form an electric current, which is then corresponding to the concentration of the biomarker that you're trying to detect. And although there are different types of electrochemical setups, like this one here is called a screen printed electrode, and this one here is called a conventional setup. So we are only focusing, and we are working with screen printed electrodes because these are highly portable and these are small and they can be easily packaged into point of care technologies, and also they are mass producible over large scale. And they can also be made on papers. They can be printed on papers and on any plastics or anything like that. That's why they're highly desirable. And basically, electrochemical biosensor has a 3-electrode system called the counter electrodes, reference electrodes, and there is working electrode, and then there is an electrolyte, and there is a potentiostat that convert the interaction between the analyte and the electrode into a signal. And these electrodes are basically, and the electrolyte convert an electrical setup, and yeah, to generate the current. Why did we choose graphene biosensors as a sensing material? Graphene is used as a sensing material in our biosensor because it has highest known electron conductivity, it has really high mechanical strength and good biocompatibility, and it has a very large specific area, almost half the area of a football pitch. And also label-free detection is possible. But when I did the literature survey of the biosensors for Alzheimer's disease biomarkers, there are not many graphene biosensors. The reason are maybe because the reproducibility of the result is not as good. And there are also issues with specificity of these biosensors. And since the on the graphene surface, the biomolecules cannot be attached directly because the pristine graphene does not have any electroactive sites to attach the biomolecules directly on the surface, and it cannot also be directly grown on the substrate. So these are the challenges that are usually present in the graphene biosensors, and that may be the reason why there are not many graphene-based biosensors, especially for the detection of beta amyloid, let's say, because that's what I've been focusing on. And this is a small portion of the literature survey that I did for the existing electrochemical methods. As you can see, there are not many. The best biosensors are here, the label-free biosensors, and none of them are based on graphene except for the one that we prepared. The reason for that, as we already discussed, there are a lot of problems with reproducibility, with the attachment of the biomolecules on the sensor surface, and so on. So basically, we only focused on 42 amyloid beta with 42 amino acid, which is A beta 42. And we detected it from the biofluids, especially human plasma and mice plasma. And then the reason is because the abnormal levels in the biofluids has been correlated with the amyloid pathology that occurs in the brain of AD patients. And the main issue with the existing technology is low sensitivity, selectivity issues. They're either the sensitivity is not enough, or the ones that have higher sensitivity, they have no specificity against the interfering species, which is really important when it comes to biofluidic analysis. And then there is lack of biofluidic analysis because most of them have not validated their sensor for any biofluids. So what is special about our biosensor? So we made this dual layer biosensor using graphene and reduced graphene oxide. Why we did that is because we wanted to combine the high sensitivity of graphene with RGO, which has high electroactive side. So the combination of two provides really high sensitivity as well as both. So it has the combination of both. And the challenges with the graphene biosensor, the electroactivity problems that can be overcome by a combination of the two systems, combining both graphene and reduced graphene oxide. And we have employed the similar structure for the detection of HIV-1 gene. And we recently published it for the detection of the protein biomarker, that is beta amyloid, in 2020. So what we did is we took a graphene screen printed electrode, and then we first step is to modify it with graphene oxide solution, which we then electrochemically reduce to produce RGO graphene dual layer electrode. Then the next step is the modification of linker. What the linker that we used here is called 1-pyrene butyric N-hydroxysuccinamide ester. It's a non-covalent linker, and the reason for using it is because it forms pi-pi stacking on the RGO. Therefore, it will not have any adverse effect on the conductivity or electroactivity of your underlying RGO or graphene structure. And the next step is the immobilization of antibodies. And then we block any non-specific binding which can occur due to the presence of functional groups on the surface using a material called BSA. It's a protein. It's bovine serum albumin. And the last step is the detection of the specific antigen, which in our case is beta amyloid. This is the characterization results of our dual layer electrode. So the first one is the Raman spectra. So if we look at the Raman spectra of graphene, it's a very standard, very generic Raman spectra of graphene. And if we calculate the ID over IG ratio, so this intensity of D over G, that came out to be 0.1. And for graphene RGO, it came out to be 1.05, which is usually very common for RGO and graphene structures. And next is the XPS spectra of, this is the overall scan. And here we can see that the intensity of oxygen in graphene is lower compared to graphene RGO structure. And the percentage of oxygen increased from 1.4 to 2.4 for graphene RGO. It's not as high as in graphene oxide structure, but that's because of the reduced number of oxygen functionalities in reduced graphene oxide. And then this is a C1S scan for graphene and graphene RGO. And here also we see an increase in the intensity of CO bonding, but not anything else like C double bond O or OCO. That is also because of the reduced number of functionalities in reduced graphene oxide. Okay, so these are the electrochemical studies that we performed with our dual layer electrode. So here we see the first one is a voltamograph for the modification of a screen print electrode with this graphene RGO. So A is graphene RGO, B is graphene and graphene oxide, and the third one is graphene. B is graphene, and then C is graphene and graphene oxide. So when you modify graphene with graphene oxide, it B decreases to C. So there is a decrease in the electrochemical response. And then when we electrochemically reduce it to form graphene RGO, then it again increases to show the maximum response. So it has high sensitivity compared to both graphene and graphene geostructures. And then on the next diagram, we have A is graphene RGO. And then B is after we modify it with linker, we get this red one here that signifies the reduction in the electrochemical response. And then when we modify it with antibody, it increases again because antibody has some available amine functional groups on its surface. So the current increases. And then when you modify it with BSA, because BSA reduces some of the functional groups and blocks some of the open sites on the sensor surface, therefore it decreases again. Okay, and this is the analytical performance of our biosensor. So these are the DPV responses for a range of concentrations that we tried, from 0.2 to 55,000 picomolar, 55 nanomolar. So we saw a very nice calibration plot between the concentration from approximately 11 picomolar to 55 nanomolar, and the limit of detection that we estimated was around 2.398 picomolar or 2.4 picomolar, which is really good because most of the beta amyloid biomarkers in blood is usually in the range of 10 to 20 picomolar in the early stages, and it keeps on decreasing as the stages progress. So in the later stages, it can go really low, in the picomolar range, or even in the femtomolar range. So this is the specificity studies that we did. So we specifically carried out the studies with ApoE and amyloid beta 40. The reason for doing this is because ApoE is present in 10,000 excess in biofluids, especially in blood plasma. And none of the studies that were conducted before have shown a specificity against ApoE or amyloid beta 40, which is really strange because right now the ratios of 40 and 42 are being discussed as a major biomarker, and for detecting the ratio, the individual detection of each of the biomarkers specifically is really important. And therefore we carried out the study with ApoE and amyloid beta 40. And as you can see, because we have a negative gradient of the calibration plot, we saw a decrease only with amyloid beta 40, and none of the the biosensors did not react to the other biomarker other than amyloid beta 42. And these are the spiked human plasma analysis. So we took some of the concentration from the linear range of the calibration plot, and we tried to plot the plot with taking the same concentration from the linear range of the biosensor and then detecting that in the spiked human plasma. And this is the curve that we got. And the sensitivity of the two were really similar, even in the spiked human plasma. And then we did some mice plasma analysis. So we took wild type samples and transgenic samples. So wild type sample is just a normal mice, and transgenic samples are the mice that are developed to express human amyloid precursor protein, which is this one, and the mutant human presenilin 1 gene. These two of the genes are associated with early onset Alzheimer's disease. And then we try to detect them using a biosensor. So as you can see, the only shift that we see, the C here represents the transgenic mice. So the transgenic mice shows a significant decrease And the other wild type mice did not show that because it does not have much beta amyloid. And we also carried out an age-based study with a mice plasma sample. So we took the samples from a 9-month-old mice, transgenic mice, and a 12-month-old transgenic mice. The sample size was around 6. And then we, with the biosensor, we can see that the normalized current shows a increase from when we go from age 9 to 12, there is an increase in the normalized current response, which estimated from our biosensor means there is a decrease in the concentration. The reason for that is because plasma levels decrease with the increase in A beta plaques burden in the brain during the post-symptomatic dementia state of AD pathology. So if you look at the brain scans, these are the immunohistochemistry results. And if you look at the brain scans, it shows that the amyloid beta plaques increase from 9 to 12-month-old, and correspondingly, the presence of beta amyloid in the blood decreases with the increase in age. So they were correlated. So what are the advantages of this type of sensor is that we use really low sample volumes. Around 20 microliter of sample is needed for diagnostics. We have a really fast analysis time. The incubation time is approximately 1 hour, and measurement time is between 3 to 5 minutes. These are highly sensitive because we have been able to achieve 2.4 picomolar for A-beta-42. They're highly specific, and there is a possibility of multiplexing. If we can attach it to a microfluidic system, proper microfluidic system, we can also do multiplexing. There's a potential for molecular diagnosis of disease, but the challenges are still associated with the reproducibility of these detection systems. And we also need to have an extensive clinical validation before we can claim that it is ready for clinical validation per se, these type of biosensors. In the end, I would like to acknowledge our funders and all the researchers, and thank you for your attention. Any questions?
Thank you, Jagriti, for your nice presentation. We have few questions from the audience.
Did you also test for other interferences in blood?
Yeah.
We checked, the 2 biomarkers that are really prominent in Alzheimer's disease, that is ApoE and interfering A-beta species like amyloid beta 40 are the 2 that we tried. We haven't tried with anything else.
Yeah.
Few
more questions. There are few more questions. How does the functionalities and performance of graphene biomarkers get affected by temperature and pH?
Temperature, and we haven't done temperature and pH studies, and these biosensors were fabricated inside a clean room. So we had a really optimized environmental conditions, but I haven't tried them outside of the clean room. So I'm not sure about how they would react to temperature changes, to be honest.
Okay. And one more question, how is it compared with FET biosensors in terms of sensitivity? What about the
of other ions and biomolecules?
Yeah.
Actually, when we were working with graphene FET biosensors, because I've also worked with them, the major problem was the reproducibility of those sensors were coming out much lower compared to these electrochemical biosensors. I mean, I would say these electrochemical biosensors are much more reproducible compared to those. And that was a major reason why I shifted from the FET to these biosensors. Also, the FET biosensors that I have seen haven't been able to achieve this this sensitivity as I did. And also, I'm still continuing to work with these sensors, and I think if with the particular surface modifications, you can even go down to femto ranges with these sensors. And I'm not sure about the FET biosensors from that point of view.
Thank you, Jagriti, for your presentation.
You're welcome.
Jagriti, thank you very much on behalf of CNR VAT. Thank you once again
Thank you.
for
Thank
such a
you.
wonderful presentation. Good luck to you.
Thank you very much.
Thank you. Thank you. Yeah. Have a great day. Bye-bye.
You too. Bye.
Yeah.
I'm
Thank
just
you.
gonna stop sharing.
Hi.
Hello, madam. Good evening. How are you?
Naren. Yeah, fine. How are you, Naren?
Good.
So you're still there, no?
Yes,
Okay. How was Suryavansh?
he's doing well.